Namaste everyone, this is Dr. Pratibha K.S., Head Department of Biotechnology at Kelly Society's S. Nijingappa College. Today I'm going to continue the part 2, the components of the blood cells and their jobs. In that you are going to understand today about erythrocytes or red blood corpuscles. Formed elements or blood corpuscles are of the following three types: erythrocytes, leukocytes, and thrombocytes. They constitute about 45% of the blood. Let us understand first about red blood corpuscles, which are known as erythrocytes. The shape a biconcave disc with larger surface area to carry the oxygen molecule. This RBC can change shape. Here, no nucleus or cellular organelles. It contains hemoglobin. And these RBCs bound by a thin elastic semi-permeable membrane. Primary function: transport oxygen from the lungs to the cells of the body and assist with carbon dioxide removal. The cytoplasm has red respiratory pigment called hemoglobin. Okay, the red cells are produced continuously in our bone marrow from stem cells at a rate of about 2 to 3 million cells per second. Erythrocytes are relatively large microscopic cells without nuclei. Okay, let us understand in detail about red blood corpuscles. Lack intracellular organelles necessary for cellular repair, growth, division. These RBCs, which are short lifespan 120 days, okay, aged RBCs and fragile, prone to rupture. Ruptured RBCs are destroyed in spleen, and phagocytic RBCs clear the debris of that particular RBCs. These RBCs are enucleated at maturity. It means they are immature, that is with the presence of nucleus, and later they will be degenerated. Then they become enucleated at maturity. Except camel nucleated RBC, lifespan 120 days, size, that is diameter 7 to 8 microns, thickness 1 to 2 micron. Shape of RBC will be disc-like biconcave. Most numerous of all formed cells. RBC count 4.5 million in female, and in male 5 million per mm cubic per meter. Okay, increase this RBCs will be increased during regular exercise and also at high altitude. Erythropoietin. Okay, here erythropoietin, the process of production of the RBCs in the kidney and red bone marrow, more RBC productions can be seen. Folic acid and vitamin B12 assist production of red blood corpuscles. Erythropoiesis means RBC formation or production. Increase RBC count, sometimes that will be abnormal, which is known as polycythemia. Decreased RBC count, that is erythrocytopenia. Organs responsible for production of RBCs known as erythropoietic organs, and at fetal stage liver and spleen can be seen production. After birth, you can see that in the production in the red bone marrow. Okay. Now, RBCs 200 to 280 million hemoglobin molecule can be seen in one hemoglobin molecule, or in one RBC you can, in one RBC molecule you can see 200 to 280 million number of hemoglobin molecule. Okay, in one RBC, in one red blood corpuscles, 200 to 280 million hemoglobin molecule can be seen. 12 to 16 grams per 100 ml of blood hemoglobin count can be seen. In one hemoglobin, 4 oxygen molecule will be bound. Hemoglobin, that is heme, ferrous, globin means protein. 1 gram of hemoglobin, which consists of 1.34 ml of oxygen. Less hemoglobin, anemia. Hemoglobin when combined with oxygen, oxyhemoglobin. Hemoglobin when combined with carbon dioxide, carbaminohemoglobin. Hemoglobin with carbon monoxide, carboxyhemoglobin. Okay, here you can see the size of this particular diameter of the red blood corpuscles, 8 micrometer. Then here this is the surface view. Okay, play very important role in transport of oxygen. Key points to remember: biconcave disc, okay, biconcave disc with thin walls, slightly elastic to squeeze through narrow capillaries, okay. Because of this particular biconcave shape and elasticity nature, these RBCs which move very easily in the blood veins, okay, through capillaries. Because of sickle shape, what happens there? Crisis happens or obstructions happens within the blood veins. These RBCs devoid of nucleus. Cytoplasm contains hemoglobin, most numerous, that is 5 million per microliters, size of pinhead. Okay, lifespan is 120 days and phagocytized by WBC. Destroyed in spleen. That's why the spleen can also be known as graveyard or blood bank. Membrane of, why that is called as blood bank? Because that is also be produced in the spleen and destroyed also in the spleen. That's why it can also be known as graveyard or blood bank. Membrane of RBC will be lysed into heme, that is heme, porphyrin and globin, that is the amino acid perfect protein, okay. Hemoglobin further digested into hemosiderin, bilirubin and biliverdin. Whereas here ferrous rich or iron storage complex reused, that is found within the cells and appears to be complex of ferritin. An orange yellow pigment formed in the liver by the breakdown of hemoglobin and excreted in the bile that impart yellow color to plasma. Okay, that is nothing but hemosiderin. A green pigment excreted in bile, oxidized derivatives of bilirubin. Hemoglobin, we can see here the 4 heme molecules which can bind with 4 oxygen molecules and known as oxygenated hemoglobin. We can see here 2 beta chain and 2 alpha chains and iron heme can be seen here. Okay, here you can see the red blood corpuscles cells with a larger surface area which binds with the oxygen, okay, oxygen from the lungs which becomes oxyhemoglobin, then oxygen bonded with hemoglobin molecules, then oxygen released to tissue cells. Okay, here also you can see the pictorial representation of the hemoglobin, that is this is the heme and exchange of respiratory gases through RBC cells, and here you can see the picture of one RBC cells. You can see there will be a presence of many million number of hemoglobin molecule. Blood, what does it need? Vitamins and minerals keep the blood healthy. The blood cells have a definite life cycle just as all living organisms. Okay, during blood loss, what happens when the human body loses a blood through a minor wound? The platelets cause the blood to clot so that the bleeding stops. Okay, because new blood is always being made inside the bones, the body can replace the lost blood. Here you can see the erythrocyte, that is the red blood corpuscle surface view, which has the largest surface area, and this is the side view of erythrocyte, which is biconcave. And here you can see a granulocyte, right? A granulocyte, there won't be any kind of granules. This is lymphocyte and this is the monocyte. Okay, the main difference here, this will be all this granulocytes which comes under the white blood corpuscles, where you can see nucleated, okay, whereas RBCs which are enucleated. Okay, this is a circular nucleus can be seen. Here you can see kidney shape or bean shape of the nucleus in lymphocytes and monocyte. And in granulocytes, where you could find the granules, you can see eosinophils, okay, to dumbbell shape. Then basophils, which is S shape with 2 dumbbells. Okay, then you can see neutrophils, many lobes can be seen over here. Okay, neutrophils. Then this is the platelets. So all these which comes under the human blood corpuscles. Today we are going to discuss only with erythrocytes. In the next video we'll be discussing about WBCs. Okay. Now let us understand here formation of new RBCs. Ruptured cells must be replaced by new cells by a process called erythropoiesis. Okay. Secretion of the hormone erythropoietin is known as erythropoiesis. New RBCs and platelets and leukocytes are produced in the bone marrow. You can see this is the bone marrow, red bone marrow. From this, all the other cells will be regenerated. Okay, you can see here, we can call the cells which are regenerated to the red bone marrow hematopoietic stem cells, in short HSCs. These hematopoietic stem cells in turn responsible for the production of red blood cells, white blood cells and platelets. Here you can see the overall picture view of blood cell origin and production. Undifferentiated pluripotent stem cell, okay, then that will be further differentiated into myeloid stem cells, lymphoid stem cells, and from this myeloid stem cell we can see megakaryocytes, erythrocytes precursors, granulocyte precursors, monocyte precursor. Further, megakaryocytes in principle blood, they circulate in form of platelets, then erythrocytes precursors, erythrocytes, granulocytes precursors, granulocytes, like eosinophil, basophils, okay, and neutrophil. Monocyte precursor, that is monocytes. From lymphoid stem cell, lymphocytes in lymphoid tissue, that is lymphocytes. Okay, so all this which comes under the red blood cells, and here you can see all these which comes under RBCs. Okay, again one more clear picture about the blood cells: blood stem cell, myeloid stem cell and lymphoid stem cell. Myeloid stem cell responsible for red blood cells, platelets and granulocytes, whereas here lymphoid stem cells, lymphoblast responsible for production of B lymphocytes, that will be in the form of plasma cell, then lymphoblast, which also gives rise to T lymphocytes. So we have two types of lymphocytes, B lymphocyte and T lymphocytes, whereas these lymphoblasts also responsible for the process of production of NK cells, natural killer cells. So all this which comes under the white blood cells. Okay, this eosinophil, basophil, neutrophils are granulocytes, whereas here lymphocyte and monocyte which comes under the agranulocytes. Plasma cells are plasmocytes, effector B cells, lymphocytes, a fully differentiated B lymphocyte which produces antibodies. B lymphocytes produces antibody-mediated immunity or humoral-mediated immunity, whereas T lymphocytes are responsible for the cell-mediated immunity. Here you can see the red bone marrow and yellow bone marrow, which are responsible for the production of the different types of blood cells. Okay, red bone marrow cells which are responsible for production of RBCs and WBCs and also platelets. Hope you have understood today's presentation. Okay, if you like this particular video, you can share, subscribe my channel. Okay, I Dr. Pratibha K signing off for today. Stay tuned with me. I'll be back with some more videos. Okay, protect yourself and protect others. Okay, keep learning. Thank you for watching.